Greetings everyone and welcome to another Genetic Engineering and Biotechnology News webinar. Our presentation today is entitled Microflow Imaging as a Tool for Pharmaceutical Particle Analysis. I'm Jeff Bogoliskis, Technical Editor for GEN, and I'll be the moderator for today's webinar presentation. Current methods for characterizing PLGA, or polylactic coglycolic acid, microparticle size, provide limited quantitative information and often overestimate size due to the presence of clustered particles. Since the size of PLGA microparticles is of critical importance for polymer degradation and drug release rates, investigators are continually searching for techniques that provide rapid and accurate particle size measurements. Microflow imaging, or MFI, is a valuable technique to many researchers as the method is able to differentiate between fines, monospheres, and agglomerates based on particle morphology and ultimately quantify the number of volume fraction for each particle population. Let's meet our speakers for this GEM webinar, where we'll discuss current methodologies for using MFI to assess size and agglomeration of PLGA microparticles. Colin Merrifield is Product Manager at ProteinSimple. Mr. Merrifield will provide a brief introduction into ProteinSimple and how many of its products are integral to quality MFI analysis techniques. Miranda Van Beers is currently a scientist in analytical method development at Dr. Reddy's Research and Development in the Netherlands. Dr. Van Beers will guide us through some of her current research using MFI for PGLA particle analysis and how the method can be employed during drug development. So before we get rolling into Colin's presentation, I want to encourage the audience to submit questions for our Q&A session at the end of the talks. We'll try to get to as many questions as we can, so simply type your question into the Q&A box on the left-hand side of your screen and hit submit. All right, with all of that out of the way, let's get our webinar started. Colin, take it away.
Good morning. My name is Colin Merrifield, and I'm the Product Manager for Microflow Imaging at ProteinSimple. Thank you for joining Dr. Van Beers and myself for this webinar on microflow imaging for pharmaceutical analysis. I'd like to start by introducing our parent company, Bio-Techne. Bio-Techne is a life sciences and clinical diagnostics company that aims to provide innovative solutions for reagents, assays, platforms, custom manufacturing, and testing services. We're proud to serve a wide range of institutions and companies who are pushing back the frontiers of science and bringing therapies and cures to patients the world over. You can see that there are three main divisions within Bio-Techne, and ProteinSimple makes up the protein platform division of the company. And at Bio-Techne, proteins are really the core of our business. We have the largest offering of antibodies in the world through our R&D Systems and Novus brands, and we've combined that analytical power with the tools that we produce here in San Jose at ProteinSimple. Proteins, and perhaps more accurately, biology, brings a level of complexity to modern research. This slide illustrates the evolution of the pharmaceutical field, where we've moved from the relative simplicity of the aspirin molecule to the complex yet very powerful monoclonal antibody. And that evolution has really ushered in a new age of research, but it's also brought a number of challenges to the field with it. These are the challenges that we've set out to solve at ProteinSimple. Research techniques are often slow, difficult to perform, require a lot of sample, and sometimes don't give you the data quality that you need. Our philosophy of innovation is to take the tools and methods that power protein research and simplify them so you get the most out of your time, effort, and your samples. This approach is embodied by our instruments, which provide answers to some of the common questions in biology. We've brought this approach to ELISA, Western blotting, imaging, capillary electrophoresis, and of course particle analysis in platforms that are easy to use, quick, and robust. And these serve a wide range of research areas, including basic R&D as well as pharmaceutical development and manufacturing. Microflow imaging has brought this philosophy and these capabilities to the particle analysis field by combining the capabilities of digital microscopy with microfluidics. Our MFI 5000 series has given researchers new insight into the particle content of their products, and our Bot1 autosampler has given them a level of throughput which really wasn't possible in particle analysis before. MFI's core competency is the subvisible size range, and these include particles that are of interest to regulatory agencies because they're common indicators of product safety and stability, but you can also find a number of other interesting particles in this size range, and these include cells, if you'd like to consider them particles, as well as engineered microparticles, microcarriers for cell culture, and the encapsulations that Dr. Van Beers has worked to develop. MFI has brought a new level of information to the analysis of subvisible particles by providing image-based analysis. So this is a sensitive approach which allows us to take pictures of particles and then quantify morphological information that can be associated with that picture. So we can measure things such as the size, shape, and translucence or opacity of the particle in order to help differentiate different subpopulations of particles within the sample. One of the classic examples of this is differentiating protein aggregates from silicon oil in parenteral products. But as Dr. Van Beers is going to illustrate, this can be applied to other microparticles as well. The Bot1 liquid handling system also serves to augment these capabilities by taking away the hands-on aspect of the work, which helps to free up users to do something more meaningful rather than run the system. And also it allows the system to accommodate a lot more samples and a higher level of throughput. This also helps to minimize the amount of sample needed for the work and make sure that the method is highly reproducible. These capabilities have helped researchers overcome a number of challenges in the particle analysis field. One in particular I'd like to highlight, which is the ability to see beyond just the size and count of the particles. The image-based approach that MFI offers and the morphology that it derives can be used to gain more insight into the development of the product. So in summary, MFI provides a range of solutions for pharmaceutical labs, whether it's throughput using our Bot1 autosampler for testing large matrices, the sensitivity that's needed to detect translucent particles, reducing the amount of sample volume that's needed for your analysis, and accommodating the viscosity that comes with a lot of biological samples. We provide all of this with a software package that gives users the ability to do in-depth analysis of single samples and high throughput analysis for looking at trends over large populations of samples. And finally, I'd like to take a moment to say thank you to our fantastic customers. They're truly instrumental in helping us design and develop our products, and our collaborators in every sense of the word. So thank you very much.
Thanks, Colin. That was a great introduction to MFI and protein simple products that may be useful for streamlining the analysis workflows. Before we move on to Miranda's presentation, I want to remind everyone to submit questions for our Q&A session at the end of this talk. We'll try to get to as many questions as we can, so simply type your question into the Q&A box on the left-hand side of the screen and hit submit. All right, it looks like Miranda's ready to go. Take her away.
Thank you, Colin Merrifield, for explaining the role that MFI can play in the analysis of therapeutic proteins. In my part of this webinar, I will be talking about how we at Dr. Reddy's are using MFI as a tool in the pharmaceutical development of PLGA microparticles. I would like to start with briefly introducing our company. In 1995, we started under the name of Octopus in the city of Leiden in the Netherlands, and we soon became a leading company for the formulation and process development of liposomes and long-acting injectables. In 2013, we were acquired by Dr. Reddy's laboratories and transitioned into an R&D center for the development of Dr. Reddy's complex injectables. In fact, many of our products are based on particulate drug delivery systems. And these type of products really need in-depth particle characterization techniques. Most of our products fall in the same size range as unwanted particles in protein pharmaceuticals. And for this reason, we started a collaboration with the group of Professor Wim Jiskoot at Leiden University in the Netherlands, who's an expert in the field of therapeutic protein formulation. Together with Leiden University, we started exploring several particle characterization techniques that were recently established for protein pharmaceuticals. MFI proved to be particularly useful in measuring our PLGA microparticle products. We recently published our results, and in this webinar I would like to share with you some of the highlights and ins and outs of this publication. What I will discuss in this presentation is that MFI can be an excellent tool for process development of particulate drug products that fall in the micrometer size range. It has several advantages over the more routinely used techniques of optical microscopy and laser diffraction. And this is because MFI is able to characterize individual particles and to measure thousands of particles per sample. This enables us to distinguish different particle populations and quantify these populations. Moreover, it is a high throughput technique, particularly when using the BOT1 autosampler like we do in our company. Hopefully, I will make all of those points clear in the next 20 minutes of this webinar. I will start with showing you a unique side-by-side comparison between MFI and Mastersizer, which is a laser diffraction instrument for two commercial PLGA microparticle products, namely Risperdal Consta and Sandostatin LAR. Secondly, I will show you how we perform particle classification with the help of the MFI software. And finally, I will show you two case studies of how MFI can be applied in the pharmaceutical development of PLGA microparticles. The first case study is about the process development of PLGA microparticles, and the second case study is about the physical stability of Sandostatin LAR after reconstitution. The two commercial PLGA microparticle products that we characterized are Risperdal Consta and Sandostatin LAR. Risperdal Consta has been on the market since 2003. It has a 75 to 25 lactic to glycolic acid ratio and a particle size of approximately 100 micrometer. The particles are loaded with Risperdal, which is a small hydrophobic molecule that is used to treat schizophrenia. PLGA microparticles containing Risperdal are administered intramuscularly every 2 weeks, whereas the non-encapsulated drug needs to be administered orally every day. So encapsulation really prolongs its therapeutic effect. And the same holds true for Sandostatin LAR, which has been on the market since 1988. It has a 50 to 50 lactic to glycolic acid ratio and a particle size of approximately 50 micrometer. The particles are loaded with octreotide acetate, which is the acetate salt of a cyclic octapeptide that is used to treat acromegaly. PLGA microparticles containing octreotide acetate are administered intramuscularly every 4 weeks, whereas the non-encapsulated drug needs to be administered subcutaneously or intravenously 2 to 4 times daily. It is clear that encapsulation of a drug in PLGA microparticles can drastically prolong its therapeutic effect. So what are the factors that influence the drug release kinetics of PLGA microparticles? Well, some of the main factors are listed on this slide. They can roughly be categorized into 3 groups, namely material properties, process parameters, and operating conditions. These all have their influence on microparticle characteristics, namely particle size, size distribution, particle shape, surface morphology, inner structure, and also drug encapsulation efficiency, which directly influence drug release kinetics. In this presentation, I would like to focus on the first three microparticle characteristics, which are related to size and shape. Why are size and shape so important? If we look at small particles, these have a relatively large surface area with a high amount of drug absorbed on the surface that dissolves rather quickly, resulting in a high initial burst release. Due to their small size, these particles generally show shorter release times. Large particles, on the other hand, will show a low initial burst release and longer release times. However, they may suffer from suspendability and injectability issues if the particles become too large. Then there's the case where we may have agglomerated particles. It is more difficult to predict the effect of agglomerates on drug release kinetics. Agglomerates may show a high initial burst release due to their relatively large surface area. It is also hypothesized that the acidic PLGA degradation products may be retained inside the agglomerates, resulting in autocatalysis, faster degradation, and shorter release times. Moreover, agglomerated products may definitely suffer from suspendability and injectability issues. And for these reasons, it is very important to obtain information on size, shape, and agglomeration of PLGA microparticle products. What are the current methods to analyze PLGA microparticle size? The most commonly used methods are optical microscopy and laser diffraction. Optical microscopy has the strength that it can measure particle size, perform particle counting, and look into particle shape. However, a major drawback is that it is a time-consuming and low-throughput method because it involves manual preparation of microscope slides, and a high number of slides are needed to obtain significant results. Therefore, optical microscopy provides only limited or no quantitative information. Laser diffraction, on the other hand, is based on the intensity and angle of light that is scattered from a sample in the measurement compartment. According to certain algorithms, the entire particle size distribution can be derived. This is a relatively quick measurement with a high throughput. However, a major limitation of laser diffraction is that particles are not visualized, and information on individual particles is lacking. Therefore, particle counting is not possible, and information on particle shape and agglomeration cannot be obtained. At certain points, we realized that the relatively new technique of microflow imaging may be able to combine the benefits of optical microscopy and laser diffraction because it can visualize high numbers of individual particles, making it a quantitative technique. In MFI, a digital camera automatically collects microscopic images of thousands of particles that pass through a flow cell. This technique has become well established in recent years to assess micron-sized particles in protein formulations. And the strengths of MFI are that it provides the particle size distribution, and it also gives information on particle counts and particle shape. And finally, it is a quick measurement with a high throughput. Thus, we wanted to explore the potential of MFI to quantitatively assess size, shape, and agglomeration of PLGA microparticles. And for this purpose, we compared laser diffraction with MFI for Risperdal, Consta, and Sandostatin LAR. On this slide, the size distribution results are compared between Mastersizer, or laser diffraction, and MFI for Risperdal Consta. We are reporting size distributions based on volume percentage as well as number percentage because Mastersizer is in principle a volume-based technique, whereas MFI is in principle a number-based technique. To be able to compare the results of both techniques, we transformed the Mastersizer raw data into a number-based size distribution and the MFI raw data into a volume-based size distribution, assuming perfectly spherical particles. From the graph on the left, it can be concluded that the volume size distribution measured with Mastersizer looks more or less similar to the one measured with MFI. Also, the DV50 values measured with both techniques are quite similar. We measured 89 micron with Mastersizer versus 92 micron with MFI. In contrast, the number size distributions on the right showed a remarkable difference between the two techniques. MFI showed an additional population of smaller particles less than 28 micron in the number size distribution of Risperdal Consta that was not detected with Mastersizer. And this was also reflected in the DN50 values measured with Mastersizer and MFI, which were 58 and 47 micron respectively. We found the same trend for Sandostatin LAR. Again, the volume size distributions shown on the left measured with the two techniques were rather similar, with a DV50 value of 54 micron measured with both techniques. However, the number size distributions shown on the right reveal a clear difference between the two techniques. This was reflected by the DN50 value of Sandostatin LAR, which was 42 micron measured with Mastersizer and 36 micron measured with MFI. MFI showed an additional population of smaller particles, this time less than 16 micron, in the number size distribution that was not detected by Mastersizer. We hypothesize that this may be due to the difference in measurement principle between Mastersizer and MFI. Laser diffraction depends on the volume of particles, and the signal will be dominated by larger particles, which may result in overlooking the smaller particles. MFI, on the other hand, measures individual particles and will also detect smaller particles in presence of larger ones. Another clear advantage of MFI over Mastersizer is that MFI can collect particle images of the two commercial products. By eye, one can already distinguish different particle populations. The top row shows some examples of small particles that were detected in both products. The particles in the middle row are individual microspheres with sizes that correspond with what is reported in literature. Microspheres in Risperdal Consta on the left appear less spherical and slightly more rough and irregular than the microspheres in Sandostatin LAR on the right, which may reflect differences in formulation and process parameters between the two products. The bottom row shows agglomerated particles. Risperdal Consta agglomerates generally consisted of 2 or 3 PLGA microparticles that were slightly misshaped, whereas Sandostatin LAR agglomerates consisted of 2, 3 or multiple PLGA microparticles that were nearly spherical. We can take this type of particle classification one step further with the help of the MFI software. One of the parameters that the software reports is the aspect ratio of each particle. The aspect ratio is the ratio of the minor axis length over the major axis length, and a particle with the shape of a needle will have an aspect ratio that is close to 0, while a perfect circle will have an aspect ratio of exactly 1. The MFI analysis software can be used to prepare scatter plots depicting the aspect ratio of all particles in one measurement. The x-axis shows the ECD, or equivalent circular diameter, which is the particle diameter that is reported by MFI. Based on aspect ratios and ECD, all particles can be classified into 3 different populations, namely fines, monospheres and agglomerates. We define fines as particles smaller than 28 micron in case of Risperdal Consta and smaller than 16 micron in case of Sandostatin LAR. Monospheres appear to be close to spherical with aspect ratios above 0.85 for Risperdal Consta and above 0.89 for Sandostatin LAR, whereas agglomerates consisted of multiple spheres resulting in lower aspect ratios. A filter was developed in the MFI software based on the mentioned ECD and aspect ratio cutoffs to calculate the volume and number percentages of each particle population. In these graphs, the resulting volume and number-based size distributions are shown for the different particle populations in Risperdal Consta. fines did not significantly contribute to the volume size distribution depicted on the left, but only appeared in the number size distribution on the right, with a number percentage of 17%. The agglomerate percentage was high, especially in the volume size distribution. With a volume percentage of 25%, the agglomerates will likely affect the drug release kinetics of Risperdal Consta. Then we take a look at the particle size distributions of the different particle populations in Sandostatin LAR. Again, fines did not significantly contribute to the volume size distribution depicted on the left, but only appeared in the number size distributions on the right, with a number percentage of 37%. The volume percentage of agglomerates was high, namely 31%, which will likely affect the drug release kinetics of Sandostatin LAR. This brings me to the first of two case studies that will show examples of what we can do with the information provided by MFI. In this first case study, MFI helped us to monitor particle size and agglomeration during the production process of a PLGA microparticle product. One batch of PLGA microparticles loaded with drugs was prepared in-house with an oil and water solvent extraction method. Samples were collected at 3 different stages during the production process and measured offline with MFI. The first sample was taken at step 1, the emulsification step. At this stage, the emulsion droplets are formed, which are the precursors of microparticles. Interestingly, only one particle population was present, namely monospheres, and no fines or agglomerates could be detected. An explanation for the absence of agglomerates may be that if one emulsion droplet encounters another droplet, they will easily merge to form a larger droplet. The second step of the production process consisted of solvent extraction and particle collection over a 25 micrometer sieve. At this stage, solvent extraction caused the emulsion droplets to solidify into microparticles. What we can see from the graph is that sieving removed many particles smaller than 25 micrometer, but not all of them. The efficiency of sieving may not have been 100% due to adherence of small particles to the surface of larger particles. This sieving step probably also explains the presence of a fines population in the 2 commercial PLGA microparticle products. The third and final step of the process consisted of drying of the microparticles. In the reconstituted final product, we see a dramatic increase in the number percentage of fines, and the reason for this may be that additional fines could have been released from the larger particles during the final drying step. Also, the volume percentage of agglomerates increased dramatically. And with this knowledge, one may be able to adjust the process parameters in order to prevent agglomeration. For instance, the use of an aqueous solution of mannitol during drying may prevent agglomeration of PLGA microparticles. In the second case study, we explored the use of MFI to monitor the physical stability of Sandostatin LAR after reconstitution. There's one known issue with Sandostatin LAR, which is addressed in the prescribing information, namely Sandostatin LAR depot must be administered immediately after mixing. And this is because the product has a high tendency to agglomerate already within a few minutes after reconstitution. Agglomeration leads to issues with injectability, and often the product cannot be administered to the patient anymore, leading to high costs due to drug loss. For our second case study, we reconstituted the commercial product with the provided diluent. One portion was incubated at 20 degrees, while another portion was incubated at 25 degrees. After 0, 4, 24 and 52 hours of incubation, aliquots were removed for MFI analysis to monitor the volume percentage of monospheres and agglomerates in time. We concluded that monospheres disappeared over time, while agglomerates were formed when the reconstituted product was incubated at 25 degrees. And remarkably, no agglomeration occurred after reconstitution and incubation at 20 degrees. So these two case studies showed that MFI provides insights into the effect of different process steps on microparticle characteristics, as well as the extent of microparticle agglomeration after reconstitution. In conclusion, MFI can be an excellent tool to assess size and agglomeration of PLGA microparticles. We have seen that different particle populations can be detected and quantified based on size and shape, namely fines, monospheres and agglomerates. On a more general note, MFI may be highly useful in the formulation and process development of various particular drug products, as long as they fall in a microscopic size range. And this brings me to the end of this webinar. I would like to say a big thank you to my colleagues at Dr. Reddy's and at Leiden University for making this work possible. I would like to thank you for your attention. If there are any questions, I would be happy to address them. I will be available as a panelist during the upcoming Q&A session, together with my colleague Kees Sloten, who's an analytical tech lead at Dr. Reddy's R&D in Leiden. And Kees is one of the co-authors and has performed many of the MFI measurements for our publication. Of course, you may also contact us anytime after the webinar by email.
Thanks, Miranda. That was a great talk. I think our audience now has a greater appreciation for the power of MFI as a particle analysis technique and its usefulness in the drug development process. So before we start the Q&A session, I want to remind everyone once again that this is their final chance to submit questions for our speakers. So hurry up and send them in now. Let's begin the Q&A. I see we have a bunch of great questions that have already come in. So let's get rolling. Give us a few moments on our side to get all the logistics straightened, and we'll begin the Q&A presently.
All right, everyone, so let's begin the Q&A. Our first question is for Miranda. Miranda, one of our audience members would like to know, they state that in the webinar you showed that MFI has major advantages over optical microscopy and laser diffraction for the characterization of PLGA microparticles. Could you also share with us some limitations of MFI?
Yes, sure. Of course, that's a very valid question. In my presentation, I've highlighted the key advantages of MFI, but indeed MFI also has its limitations. The downside, for example, of detecting particles one by one is that MFI results can be much more affected by the presence of only one particle that deviates from the rest. So especially the volume size distribution can be affected by the presence of just a few larger particles. So you really have generally higher standard deviations at the larger size bins than, for example, with Mastersizer. So if you look at the Mastersizer curves, they look really smooth and they are averaged, nice and smooth curves, which you may have seen in my presentation. But yeah, the upside of this is that MFI has a much higher sensitivity towards sample-to-sample or batch-to-batch variations, which can be an advantage, of course, for pharmaceutical development and quality control. So another difference I would like to mention is that MFI can measure in a more limited size range. So this Mastersizer instrument that we have been using, it measures in the size range of 2 micrometer up to 300 micrometer, whereas the Mastersizer measures in the size range of 20 nanometer up to 2 millimeters. So size range is definitely a limitation, but if your product falls in the right size range, it works perfect. Another disadvantage of MFI I would like to mention is that you really need to dilute your sample quite a bit in order to prevent this coincidence of particles. If you use a too high concentration, then the particles will be located really close to each other, and the MFI will detect these two particles as one particle. So you really need to dilute your sample. Well, Mastersizer samples are also diluted, but definitely less. Furthermore, maybe good to mention is the compatibility of MFI with organic solvents is limited, while Mastersizer, for example, can handle a wide range of solvents. Mastersizer can also measure dry samples, while MFI can only assess particles in suspension. So these are really things you need to keep in mind. And maybe one last point I would like to mention is that if you would like to obtain additional information besides size distribution, such as agglomerate percentage with the MFI, then the MFI data processing is rather time-consuming and really requires a certain level of expertise. But of course, you do get the extra information, and you may also use the help of external software, such as the open source software of R, ImageJ, to process MFI image files. But yeah, this is also a point to keep in mind. Yeah, so hopefully this answers the question.
All right, thank you very much, Miranda. Colin, we have a question for you. One of our audience members would like to know, what is the smallest particle size for which MFI can be used?
Yeah, so we have two different MFI models. There's the MFI 5100, which Miranda used in her study, which analyzes particles in the 2 to 300 micron size range. We also have the MFI 5200, which analyzes particles in the 1 to 70 size range. So that's the more sensitive model, and the lower limit of that is 1 micron.
All righty, thank you, Colin. Miranda, another question for you, and possibly for Case as well. How did you settle on an aspect ratio for your morphological characterization of the particles?
This question will I answer, Colin. We use the filter manager in the View Analysis Suite software, where we select the ECD, so the equivalent circular diameter, and the aspect ratio. And we set up the aspect ratio over the circularity, because we were able with this aspect ratio to distinguish, to have more power to distinguish different particle size populations like fines and monomers and agglomerates. And that's the reason why we took the aspect ratio over circularity to have more power to separate between the populations. I hope this will answer the question.
Thanks very much, Case. Miranda, another question for you guys. This is a question concerning the fines population that is dedicated to the MFI but not with the Mastersizer. How do you know that these fines detected with MFI are not artifacts or contaminants that appear during sample preparation or MFI measurement?
All right, I tried to answer this question. Indeed, we were also wondering about the nature of these fines when we first observed them with MFI. And of course, the fines could be extrinsic, so they could be contaminants, or intrinsic, related to the drug product, or inherent, and that is originating from the drug product. We cannot exclude that some of the fines consisted of extrinsic particles, such as dust or hair fragments that contaminated the sample during sample preparation or MFI measurements. But as a control, the baseline measurements were performed with sample diluents, and these generally showed particle concentrations that were 5 to 10 times lower than the fines population in commercial PLGA microparticle samples. Thus, most likely mainly intrinsic or inherent particles were present in the fines population. And indeed, we found an explanation for the presence of the fines during our first case study, as I showed during the presentation, in which we studied the effect of different process steps on PLGA microparticle characteristics. So it's very likely that the presence of the fines is the result of collecting the solidified particles over a sieve during the production process of PLGA microparticles. And from the number size distributions before and after sieving, it really looks like the fines were part of the original size distribution before sieving, and then sieving reduced the number of fines and contributed to the gap in the size distribution. So MFI really provided this information. I hope this answers the question.
Thanks, Miranda. Colin, we have one more question for you. One of our audience members would like to know, what is the MFI data size per sample?
So for the samples that Miranda was analyzing, as she mentioned, they're samples that have a lot of particles in them. I would expect the size of those analysis files to be somewhere in the 200 to 300 megabyte size range. So they're relatively large because because of all the image files that MFI captures during its analysis.
Alrighty, thank you, Colin. And it looks like with that, we've come to the end of our webinar. So I want to remind everyone that the webinar will be archived for up to a year on our website at www.genengnews.com. So if you missed parts of it, you can watch it again, or feel free to link to your friends and colleagues so they can watch it. I'd like to thank Colin and Miranda again for their informative presentations, and thanks to Case for joining in the Q&A session. And I'd like to thank the audience for their attention and thoughtful questions. And a very special thanks to Protein Simple for sponsoring this webinar. So hopefully we'll see you again at another Gen webinar in the near future. Goodbye for now.